Tamara Nicholson. Welcome to Marrone Bioinnovations. Come on in.
Hi, my name is PEARSON. I am the Invertebrate Pest Biology Group Leader here at Marrone Bioinnovations. That's just a really fancy way of saying that I play with bugs and all the creepy crawlies. So here at MBI, we develop bioinsecticides or biopesticides, and those are used to help control different pest species out in agricultural settings, and we usually use kind of like bacteria or fungi to help us produce our products. And so luckily here in entomology, where we deal with insects, I get to be part of that entire process. So that is when we are discovering a new potential product. So that'd be like an early stage where we're trying to figure out what microbes might be working and what fungi might be working for us. Moving that into some sort of development after we've discovered some of the activity that we like. Helping to further advance the product by understanding a little bit of how it might be working and how it might be controlling these insects or bugs. And then also helping with the product release and making sure that our product is of a certain quality and is reproducible every time so that our end customer gets the best possible product. So how do we do that? That's an excellent question. If you follow me this way, I'll kind of show you how that might be done. So here we have an example of some of our bioassays that we use in-house. And these bioassays can use a variety of different insect species, a variety of different critters that we can use across these different platforms. And so some of them, as you can see here, actually use plant material. So here we have a cut pepper leaf, and we'll actually release aphids on there. These leaves can be treated, and we can look for things like mortality, so that's kind of a hard and fast tell. We can also monitor how many young our offspring these aphids are having, and that can let us know how we might be affecting subsequent generations. We also have artificial systems that are completely artificial that don't have any plant material in them. So this is another example of one of our assays. So we actually have this little diet pack here, and that's actually got a treatment in it. You can kind of see it there, it's a liquid. We mix this with an artificial diet that these are actually little ligus bugs. I don't know if we can kind of see those. If I tilt that up, maybe a little better. And so these guys are a piercing, sucking insect. So that means their mouth parts are kind of like a little straw. So they actually will insert that into this diet packet and start drinking up the slurry that's in there. And we can see how that might be affecting them as well. These assays are what we wouldn't exactly call high throughput. So if we want to study a lot of things very quickly and we want to learn how something might be happening, we probably go to something even more smaller or compact. So what we have here is another artificial system, and this is actually an artificial insect diet that's pipetted out into each one of these little individual wells on this 96 well plate. And this allows us to look at a lot of different treatments all at once. So this really speeds up the process for us to help us understand what might be going on. So we do have a variety of different assays that can look at different things and let us know what's going on. But the other aspect that's very important for us is to make sure that we have insects, right? So we want to make sure that our colonies are available all year long for us to do our testing and help us understand what might be going on with these microbes. So if you follow me in here, we'll show you kind of briefly how we actually rear some of these. So this is one of our insect rearing rooms. It's a little loud in here, sorry, so I'm going to try and speak up so hopefully you hear me on the video. Apologize if it's too loud. But this is actually our incubators here. This is where you can keep things at a constant temperature all year round. You can also control your light schedule. So your lights can be on and off. So in nature there is a natural light and dark period. So we can set these for different times to represent maybe different parts of the country or different times of the year and so on and so forth. So that can all be replicated here in one of these types of chambers. And if we look at what we have here, this is actually that same artificial diet I was talking to you about earlier. You can find this here, this giant cube of it there. And what we have next to it are the insect eggs. So on this paper, all those little dots you can kind of see there, those are all the insect eggs. And these are actually going to be for a caterpillar or a little cabbage looper in this case, happens to be the species here. And these will hatch out into little worms. So we can kind of see that as we move through their life cycle. And like what we talked about earlier, those were a piercing, sucking insect species, so they have the straw-like mouth parts where they're drinking. These guys are just chewing. So there's the ones that, if you see a big hole in your leaf out in your yard, more than likely somebody like these guys is hanging around, causing some problems potentially. So this is what they look like when they get a little bit older. And you can kind of see, so this again is that same diet. It's used over and over again for different types of applications. And these are a little bit larger caterpillars, something you might see in your garden, for example. And these guys will continue to grow, and they will go through their last change, or their last metamorphosis, where they'll actually form a little cocoon and turn into an adult moth in this case. So here, let's see if we have, oh, we don't have it in there, so we'll take you in to see the adult moths here. Thought we might have some debate. Keep following me this way. So we'll come around to this side, and we'll actually show you what these guys look like as adults. So let's see if I can take this over here, open this up, show you here. So these are all the adult moths, right? So they're not exactly spectacular. They're just a regular kind of brown moth you might see anywhere, hanging around, flying around, hanging around your light at night on your front porch. But these guys are actually, some of these species are actually detrimental to agriculture, and so they're the ones we target to help control. And so what we do in these adult cages, we put that paper you can kind of see hanging down in there. That's actually going to collect their eggs, and so then we collect their eggs, we wash them, and the whole process starts all over again. And this is just one of a whole multitude of different species that we can possibly keep here at MBI that allow us to better understand how current and potential products might be working.
Hi, I'm David Castillo. I work here at Marrone Bio as an associate quality control scientist. And what I do here is I do testing for various products that Marrone Bio has. The products we have are, I have a little array here for you guys. This is 206. It is a insecticide. I have here some granule pellets, 203, also an insecticide. I have 401 TJI here, some lesticide. And what I do, and what my team members do as well, is we take these products and we test them for physical properties. So we check things like pH, percent moisture, viscosity, dispersion, which is actually this test right here. And I can do a little demo for you guys. So what we do is we're testing how this product here, 106, disperses when a farmer mixes the solution. So for example, this is the end result. We want it to be uniform. So to test how it disperses, I just get a few drops, come here, add the drops to the liquid, and I see how it disperses. I want to make sure it's all uniform here, and disperses good, so when the farmers mix it, it's easy for them to mix it and spray it in their sprayers. We have other things, pieces of equipment here. We have our pH meter. We have our densitometer here. It's a cool piece of equipment. It tests density. Overall, quality is very important for the company. We make sure that our products are good for our customers, and that's why it's really important to have a good, strong quality department.
Hi, my name is Dr. Brittany NORDMARK, and I am the Formulations Group Leader here at Marrone Bio, and today I'm going to be showing you the Formulations Laboratory. So come this way. This is our main laboratory where we do a lot of our small sample work and benchtop work. So not a lot of equipment in here, but we do have basic things such as pipetters, scales, balances, etc., things like that. But if we come over to this lab, this is where we do a lot of our analytical work. So over here we have a particle sizer, which tells us just what it sounds, how big particles are in the formulation. Over here we have a viscometer, which measures the viscosity of the formulation. And then over here we have an HPLC, which stands for high pressure liquid chromatography. And with this machine, we use it to trace our actives in our formulations and keep track of them over time to make sure that they remain stable. One more lab to show you guys, if you come this way. this is our manufacturing laboratory. If you look over here, this is our water dispersible granule setup. So in order to make a water dispersible granule, you first start with a mixer, kind of like you do in a kitchen. You go ahead and make a dough, and you feel it to make sure that it has the right sort of moisture content. Once you have the right type of dough, you go ahead and put it through an extruder, which is disassembled at the moment, but it comes out like spaghetti. Like when you're a kid playing with Play-Doh, it kind of looks like that. You then take this spaghetti formulation and stick it in a fluidized bed dryer, which basically just blows hot pressurized air and dries it within a few minutes. And the resulting formulation is this water dispersible granule, which just looks like a bunch of little cylinders. We also have a spray dryer, which is used for taking a liquid and turning it into a powder. This is a benchtop spray dryer, but we also have a pilot-sized spray dryer, which is over here in this room. I'll go ahead and let you. Yeah. This is just a very large spray dryer, about, I believe, 12 feet. How it works is that you put your formulation into this feed tank. It then goes through this pump and goes all the way up to the top, where it goes through a nozzle. When the liquid goes through the nozzle, it gets sprayed into fine particles and falls through this chamber, which is very hot. And as the particles come through the hot chamber, they dry into a powder, also disassembled right now. But there would be a tube down here that would lead up into this cylinder or cyclone tube, and then down into a container that would be here, and we would collect our powder material. And that is everything that I have to show you here at the Formulations Lab. Thanks for listening.
Hi there, my name is Jacob BLODGETT. I work in the Bioprocess Technologies department, where we work on growing the microbes that we receive. First off in small-scale fermentations like this 5-liter fermenter here, as well as the 10-liter fermenter right here. We work initially here on figuring out what the microbe likes, what it doesn't like, and figuring out how to optimize the production of the compounds that we want in order to produce quality products. And then from here, once we move on, once we've optimized at this stage, we then start to work on growing at larger scales, which we'll see in the next room. Okay, so once we have optimized how we like to grow our microbe with whatever nutrients it requires, we then move on to a larger scale tank like this one, which is a 20-liter fermenter. Here is where we start to finally play around with either back pressure or the actual tank size in order to control for any sensitivities that the microbe might have as you scale up in size. And so this 20-liter fermentation is the convenient transition between going from a 10-liter glass fermenter to what will be the 100-liter fermenter right over here in the corner. Now we have 100-liter fermenter right here, 100-liter fermenter here in the corner, and then there's another 50-liter one right over here in this other corner. These are where we start to optimize, kind of finalize the small details of what the fermentation needs to achieve before we eventually scale up to our manufacturing plants, whether they be in Michigan or wherever else in the world. Here's an example of a fermentation that we have running now, similar to what was seen in the glass fermenter lab. However, this one is a little bit more complex. We kind of start to optimize some of our automation a bit more. We start to control with more intense feeding operations, and then unlike the glass fermenters, the steel fermenters are held in place and are sterilized with the utilities attached to the building, like steam and whatnot.
Hi, my name is RATNAKAR ASOLKAR. I'm the group leader for chemistry here at Marrone Innovations. As you might be knowing, at Marrone we discover, develop and manufacture biopesticide using natural source. And what we do here is chemistry. Is we go through this process to identify what are the compounds which make our product potent and different from other competitors. So the process starts from isolation of this microbes or the selection of the plant source all the way to extraction, bioassay, and identify the active. And the role of the most important work of the chemistry is to find out, identify these compounds which make this product potent and it help to protect the crops. So we starting with the project, we start with the source, either the microbes, extract them, do the bioassay, and confirm the activity what we see in the preliminary assay. And then we move on to the next step, which we do extraction, fractionations, and so on and so forth. So here are some, we have some machine here. Once we know the compound, the fractions are active, we try to purify them using HPLC, preparatory HPLC. We purify them, after purifying, we confirm the activities, and then we do the NMR and assign the structures. Once we assign the structures, we develop a method, and the method is used to quantify what amount of the compound is present in the compound in the sample. And once the method is developed, we transfer the method, validate the method, and then later on this information is used by other groups like fermentation to optimize the yield, formulation to stabilize the product based on the active compound. And then once the method is finally established, we validate the method, then transfer to the QC so that QC use it, and then they use that method and release the product for chemistry purpose, as well as we transfer the method to our contract lab for like doing the tox study. They use these informations and then complete the study. So in short, my group work on mostly on isolating the compound and then identify, develop methods, and that helps the other group to use that one and make a potent product. Thank you.
Hi, my name is Brittany Pierce. I'm the group leader for the microbiology department at Marrone Innovations. And in the microbiology department, we do everything from early stage screening to understanding how our products impact plants and the microbiome and soil health communities. So to give an example of some of the microbes we work with, you see them on the plates here. We work with a variety of microbes, both gram-positive and gram-negative. And we also work with some fungi. So a lot of the early stage screening would be based on growing these microbes and testing them in various types of bioassays. Some of the microbes are tested against different types of insects, some are tested against fungi, bacteria, and then some of them are used for plant health. Once a microbe has been developed into a product, we then try to understand more about the active compounds that microbe produces, as well as the way that the microbes interact with the plants. So we use real live plants, and we apply the microbes, and we look at the way that the plants respond to the presence of those microbes. So a lot of times we'll be looking at responses in the leaf tissue. We also look within the soil, especially in the rhizosphere, where microbes will work in close association with plants to help the plants increase their tolerance to stress and acquire more nutrients. And in some of this, we've done a lot of work with a product called the Noble, which is a really cool microbe that can help kill bad bacteria in the soil and bad fungi. But it also helps retain diversity that you can see in those graphs there. And the diversity is really important for a healthy soil community and sustainable agriculture. Beyond this, we work a lot, like I said, in the plants and trying to understand how microbes interact with various stress pathways. Beneficial microbes can help a plant acquire nutrients that are otherwise unavailable to them in the soil. It can help them increase their ability to uptake those nutrients, utilize them more efficiently, as well as respond to different hormonal and defense pathways. And these can provide a lot of benefits to the plants and reduce the pesticide applications that have to be used.
I am Lucho, and we're standing here in a research research greenhouse that belongs to Marrone Bioinnovations. So this is a little bit different than a greenhouse that is used for raising crops. And we don't really harvest anything in here. What we do is we plant lots of things in order for them to be tested later on. So, for example, we have some radishes here, we have some peas, we have some cucumbers on their way, we have some corn. So all of these things we'll use for different kinds of tests for our different products. So if we have a fungicide, we'll grab the cucumbers, we'll take them out of here when they're big enough, we'll put them somewhere else, put some disease on them, and then we'll try our product on them, make sure the product is working the way we want it to, or try to tweak our product. Yeah, and that's kind of what we do at a research greenhouse. So here are some plants that are inside domes. They're all covered up because what we normally do, wherever you have plants, any greenhouse, any farm, any garden, you have pests. They immediately appear, and the way we control the pests in this greenhouse is we release natural predators. So we buy these, we get them in the mail every couple of weeks. We leave them open and the eggs hatch, and the little tiny wasps, they're almost invisible, fly around and eat the aphids or whatever it is that's bothering our plants. But since we have insecticides and we need to test the insecticides, if we get a plant to test the insecticides that already has wasps on it, it's going to mess up the test. So sometimes we have to grow plants protected from the wasps in these little domes, and that's what's going on here. So we have to kind of put our hand in and water it and then tie it back up so that nothing gets in here. Here we have some plants that were brought over from the greenhouse once they grew big enough. And these are cucumber plants. And if you notice here, they're full of white powder. This is called powdery mildew. And that's a common disease that happens on lots of different types of crops, but cucumbers particularly susceptible. So we have, it likes it very humid, so that's why it's behind these plastic sheets. So the moisture stays in there, they're sitting in a little bit of water. So in order to run our fungicide test, we have to be able to have fungus growing on plants. And this type of fungus won't grow in a petri dish, it has to grow on a plant. It's sort of an obligate parasite, an obligate disease. So we use these plants in order to test different types of treatments with our fungicide. If we want to combine it with something else, if we want to change the rates, all sorts of questions that come up around the product. So one of the types of tests that we run is a crop safety test. Most products that you put on plants, if you put them on at the wrong rate, if you mix them with the wrong thing, they can actually cause damage to the crop. So we want to make sure that our products never cause any damage to the crops. We'd feel really bad, and so would the farmers. So we test them, and anything that we make, we run it through tests here. So you have different species of plants here. You have cucumber, you have spinach, tomatoes. So here we have a positive control, which means we got something that we know does damage to the crop, and you can see that's pretty ugly looking spinach. And then we have that as sort of a reference, and then we test our product, and our product comes out looking really nice. So we're happy. We can release it, take it to market, and sell it.